Observe the wild agapornis in its natural habitat, the standard green lovebird that serves as the baseline for the entire species. Modern aviculture has moved far beyond this single hue, producing a spectrum of violet, Lutino, Dutch blue, and sea green variants through generations of selective breeding. Producing these variations is not a matter of mixing colors like paint. It requires a practitioner to work within the rigid frameworks of Mendelian genetics. This process carries significant physical stakes. If a female is bred before she is mature, she may lack the calcium reserves needed for shell formation, leading to dystocia, or egg binding, where the egg becomes stuck in the reproductive tract. Beyond the mother's health, improper pairings can activate lethal recessive mutations. Genes like stumpy limb or hereditary multiple malformation often result in embryo death before the birds can ever hatch. To reliably produce a healthy, rare color mutation, a breeder must manage both these invisible genetic risks and precise environmental variables. The logic of these mutations begins with a fundamental difference between avian and mammalian genetics. In birds, the sex chromosomes are reversed. While human males are XY, male birds are ZZ. It is the female who is heterozygous, possessing a Z and a W chromosome. This ZZ-ZW system is the key to sex-linked traits. Mutations for colors like Lutino or pallid are carried exclusively on the Z chromosome. Because the female only has one Z chromosome, she only needs a single copy of the gene to show the color visually. A male, however, must inherit the mutation from both parents to express it, otherwise he simply carries the gene internally. Breeders use this chromosomal math to predict exactly which pairings yield specific colors, targeting rare mutations without the risks of blind inbreeding. While the genetic code provides the blueprint, the physical environment acts as the trigger for the breeding cycle to begin. Age is the first safety threshold. Birds should be at least 12 to 24 months old, ensuring the female has the muscular tone and nutrient reserves to handle egg production. Environmental cues signal the shift. 10 to 12 hours of light mimics natural photoperiods, and supplying a wooden nesting box with willow or palm leaves simulates a peak breeding season. These external changes prompt the birds to initiate the reproductive and nesting behaviors required for a successful clutch. The sequence begins with the female laying one egg every other day, followed by an incubation period that typically lasts between 21 and 25 days. At day 10, breeders use a technique called candling. Illuminating the egg confirms the presence of a fertile membrane and a developing vascular system. In the final hours, the chick uses a specialized egg tooth on its beak to breach the shell, cutting through the calcium barrier in a process that requires significant physical effort. The emergence of the chick marks the point where the breeder's management of genetics and husbandry successfully aligns with the bird's biological needs. Immediately after hatching, the chick relies on the remaining yolk sac, which is absorbed directly into its body cavity to provide essential nutrients for its first days of life. Socialization begins at 1 week old. By gently handling the chicks for short periods, breeders help them adapt to human interaction, which is necessary for them to become tame companions. Sustaining this lineage requires the same commitment to data seen in foundational Mendelian research. Meticulous pedigree records are the only way to track recessive traits and prevent the accumulation of genetic disorders. Responsible aviculture is a discipline that demands more than an eye for color. It requires a deep commitment to the biological health and ethical management of the species.